In this video, I'll describe the mechanical structure of DNA and also a little bit about epigenetics. So I'm talking about DNA in the nucleus, also referred to as nuclear DNA. So this is the DNA in eukaryotes, and it's packaged as chromatin, which is a complex of DNA and proteins. So the DNA gets looped around these, it's a nucleosome. I think the structure of the DNA is wrapped around histone proteins, and I think either the protein complex or the DNA plus the protein is considered the nucleosome. So this is where you get beads on a string kind of a formation, kind of the most basic structure of DNA packaging in a nucleus. So epigenetics is about tagging the DNA, but you can kind of see here DNA wrapped around histone proteins forming nucleosomes, and then those beads on a string get structured into a more compact form of DNA or chromatin, and that gets not really looped like that, but there's various other higher level formations of the chromatin, and then in mitosis or meiosis you get kind of this chromosome structure when it's duplicating the DNA or separating duplicated DNA. So epigenetics is adding various tags on the DNA itself or the histones. DNA methylation is where a methyl group gets added to the DNA. I think the most common is the 5-methyl cytosine, where the methyl group's added to the number 5 node in the cytosine base. So in the DNA, there's specifically these CPG sites where there's a cytosine followed by a guanine base. And you can see that in mammals, at least 70-80% of the CPG sites are methylated. And this is something that's preserved during DNA duplication or DNA replication. You have a CPG on one strand matched with a CPG on another strand. So if you have a methyl group on one strand and you form a new DNA strand on the other side, there's a process of adding the methyl group onto the new DNA strand. And the histones can also get methylated. The methyl group added to one of the amino acids in the histone proteins in the nucleosome. It's again, I think I mentioned it, but maybe not. The tagging is a way to control transcription of the DNA. Here with histone methylation, you can either increase or decrease transcription. And I think methylation of the DNA usually reduces transcription. And same with the histones, you can add a acetyl or acetyl group to it. And that's a way to, again, I think it increases transcription when there's an acetyl group added to the histone. The methyl group is pretty obvious. It's just a carbon with 3 hydrogens, but the acetyl is not actually listed as a functional group. It's kind of this generic form here where X is a methyl group. So you can see the acetyl group here is kind of listed as a moiety because it's a common thing, but it's a little bit more specific than the generic functional group for it. I guess the last thing to mention is just the epigenome and epigenetics. The epigenome is kind of the entire set of epigenetic markers and tags that are present in DNA throughout the entire organism. I think epigenetics kind of has two definitions. One here is heritable changes that are not in the DNA, and then here the changes themselves. So when you have a single cell starting point for a multicellular organism, you have some epigenetics there that were inherited from the parents. Sorry, I lost my train of thought there. So yeah, all the cells of the developing organism get that information, but then once there's cellular differentiation, those tags choosing which genes to express and which rates and that kind of thing are modified depending on which type of cell it is. So the 30 trillion cell human has some epigenetic information in every single cell, which is different based on what type of cell it is, even though they all share the same DNA. So the epigenome is kind of taking all that into account, or it's just the single cell at the beginning of the process.